demonstrating you immunofluorescence experiment, how it is done, and what chemicals and what reagents are used, and each and every step we will explain, and why the rationale behind each and every step. We will explain everything to you. So you can ask any questions in the chat box, and we'll answer them at the end. Okay. So let me show you the protocol of the immunofluorescence experiment. These are the basic steps that we will be following, and this is how we have prepared all the buffers. So this is according to the protocol. All of this we have prepared all the buffers, 1x PBS and spheroplastin buffer, and this is the blocking solution. And now we'll start. So we have grown from the primary culture, we have grown the secondary culture of OD 1.2. For meiotic cells, we take SPM culture, and for the mitotic cells, we take YPD culture. And we have taken 5 ml of culture from this into this 15 ml Falcon. Now, I will add, as per the protocol, I will add 142 microliter of 37% formaldehyde solution. 542 microliter. And this is the formaldehyde solution from Sigma that we take. This is 37% formaldehyde. And this will make up the final concentration of 4% once we add it into this 5 ml culture. So I've added the formaldehyde into this culture. So why we use formaldehyde? We use formaldehyde for fixing the cells. What formaldehyde does, it freezes the cells in their current state, preserving. Okay, so it preserves their morphology while maintaining it, and it also prevents proteolysis and prevent postmortem decay. Okay. So we'll take this culture, and we'll keep this, after adding formaldehyde for fixation, we'll keep this culture at room temperature for 2 hours. So at room temperature, I'll leave this culture for 2 hours, and then after 2 hours, we'll take this culture and centrifuge it. Since it's not possible for taking the time constraint, it's not possible to centrifuge it down and show you how this 2-hour incubation step is done. So we have kept these centrifuging fixed cells. This pellet is of the centrifuge and fixed cells. So you already know the rationale of fixation. Now we'll wash these cells by resuspending in 3 ml 1x PBS. So washing step includes discarding the supernatant first. So I'll discard the supernatant here. I have the small discard bin. And I'll add 3 ml of 1x PBS. I'll take this 1 ml. Mhm. So I've taken 3 ml PBS in this packet. Now I'll resuspend the pellet by either vortexing it. So this pellet has been resuspended in 1x PBS. We generally keep PBS in 4 degrees in the fridge. And now I'll centrifuge it at 2000 RPM for 2 minutes. So this is the thermocentrifuge, and it is important to balance it first. 3 ml. So we have balanced it with 3 ml just water, and this is our cell pellet, fixed cells. So we'll centrifuge it for 2 minutes. I'll start it. So this will centrifuge, and um, so we have already prepared the another culture which is centrifuge and treated with 1x PBS. So I will again discard the supernatant, and now I will add 500 microliter of spheroplastin solution. So what is the rationale behind using the spheroplastin solution or this spheroplastin buffer? That Rashmi will tell you.
Okay. So actually, spheroplasting is done to remove the cell wall from the yeast cells. This allows the antibody to have better access of the intracellular structures that would otherwise be inaccessible because of the cell wall barrier. This enables accurate utilization and localization of the target protein within the cell using the immunofluorescence technique.
So I have added 500 microliter of spheroplastin buffer to it. This spheroplastic buffer contains sorbitol, and this is important to maintain the osmotic pressure. You can either mix it with by vortexing or by just pipetting. So that's up to you. And so added 500 microliter of spheroplastin buffer into it, and so now you can keep this pellet for several days at 4 degrees Celsius, or if you want to perform the experiment, continue it that day only, you can use it then and there.
Okay, so now we will show you how the spheroplastic cells look. So is my screen visible? So this upper panel shows you the non-ferroplastic cells, and this is how the ferroplastic cells will look because their cell wall has been disintegrated. Now moving on to next step.
So I hope by now you have a clear picture in your head how these cells would actually look like, these spheroplastic cells. So I'll take the 500 microliter of these spheroplastic cells in 1.5 ml MCT, and I'll add 3.5 microliter of beta ME.
Yeah.
10% beta ME.
So beta mercaptothenol is actually a potent reducing agent which cleaves and reduces the disulfide bonds.
3.5 microliter of beta ME. So beta ME is light sensitive, hence we have kept it in foil covered falcon. I've added 3.5 microliter of beta, 10% beta ME. in this 1.5 ml MCT. And now I have to add 5 microliter of 10% zymolase. Now, zymolase is an enzyme, hence we have kept it in ice.
It is usually kept at 4 degrees. So why we use zymolase? Actually, it is an enzyme used to lyse living yeast cell walls to produce protoplast or spheroblast. Zymolase treatment is given in the presence of spheroblasting buffer because as we added it earlier, since it contains sorbitol, which is important to maintain the osmotic pressure and prevents the cell wall bursting. Hence, sorbitol, it's a osmoprotectant.
Now these cells, these are the fixed cells containing spheroplastin solution, 10% beta Me and 10 mg per ml zymolase 20T. And now we'll incubate them at 30 degrees for 45 minutes in a water bath. So usually the step is done in water bath only. So I'll keep them. Now, we have these
We already incubated the cells at 30 degrees Celsius for 45 minutes. So we will continue from the next step, which Keith will demonstrate you.
So we have centrifuged them at 2000 RPM for 2 minutes. And now, so after 30 degrees Celsius treatment of 45 minutes, now I'll discard the supernatant of these cells and again resuspend them in 200 microliter of spheroplastic buffer. 200 microliters of spheroplastic buffer. It's important to resuspend them. Now our cells are actually ready for the immunofluorescence experiment to be treated with primary and secondary antibody. You can keep them immediately in ice or just use them. So I've kept them in ice so that meanwhile I can prepare our slide on which we will put these cells. We'll keep them here and kept the enzyme in ice. Now, these are the Teflon coated 10 well slides for immunofluorescence experiment. These are the slides.
So you can see there are 10 wells.
And it's really important to wipe these slides away from all the dust. And so I wiped them with tissue paper once, kept them here. And this is a vacuum pump. Using this vacuum pump, I'll first take off all the dust which is inside these wells. So if there's any dust, you just have to move it without any water or anything. Just move the pipe on top of the wells. Not on the wells exactly, just the edges of it. So that if there's any dust, you can remove it. We just need one well, so it's fine for now. So we add 0.1% polyal lysine. This we have got from Sigma again. This is 0.1% polyal lysine, and the rationale behind using this is
Polyal lysine is used for non-specific attachment factor, is a non-specific attachment factor for cells, and it is useful for promoting cell adhesion to solid substrates by enhancing electrostatic interactions between the negatively charged ions of the cell membrane and the cell surface.
I'll take this solution, put it for some time on this well. Make sure you don't touch the well with the tip or even with your hands. Just pour anything on top without touching the tip to the well. Now we'll generally it is advised to leave this for 10 to 15 minutes, but since the time constraint, we already have these wells treated with polyol lysine, and I'll just remove this solution now. Make sure you don't touch the well from the inside, just from the edge, remove the spinaci. All the polyol lysine, it has been removed just with the vacuum suction. Now we have aspirated off all the polyol lysine, and now the well is ready for our cells to stick on it. So we'll add 20 microliter of our spheroplastid cells that we have prepared on this polyol lysine treated well. Make sure you don't touch the well with the tip. So this is the pellet of the spheroplastid cells. We have kept them in the well treated with polyol lysine, and we leave it for some time, again 5 to 10 minutes, and then aspirate it off. So we will just demonstrate you how to aspirate it off again. And so again, keeping it on the edge, aspirate it off.
Then we have to wash it.
Wash it 3 times with 1x PBS. Yeah, we have already prepared 1x PBS, and wash it 3 times using 20 microliter of 1x PBS. So we generally take 20 microliter of everything because that is the capacity of this small well here.
So I think it's visible on the slide.
There is a small drop. And when we put this pipe on the edge of this well, it's off. Right. So again, again 20 microliter of PBS. Second wash. And then we will do the third wash. So no PBS should be left in the well or in the edges. Make sure you remove everything.
You can see how vacuum pump is working. There are bubbles in the pipe.
Now we will put this slide in methanol, which is already chilled and kept in 20 degrees Celsius.
-20.
We'll take this slide to -20, the chilled methanol. So this is a bottle filled with methanol, and the another one is filled with acetone. Another one, it's filled with acetone. So in the first step, I will dip this slide in the chilled methanol. Yes. I will just keep it here. Yes, you can see that all the wells are fully dipped inside the methanol, and I will immediately keep it back and leave it for some time. So Rashmi will tell you what is the rationale behind using methanol and acetone. Why do we use it?
So actually, methanol and acetone, they both are used as fixatives. They preserve the structure of the cells from rapidly dehydrating and precipitating proteins, which allows for better antibody binding and visualization of target antigens within the cell. So actually, methanol and acetone act by dehydration and protein precipitation, thereby fixing proteins. This instantly permeabilizes the cells.
Just like I showed you, I demonstrated you how we dipped it in chilled methanol. We have to keep it for 5 to 10 minutes again. And then immediately from methanol, we dip it into the acetone bottle. And again, we leave it for 5 to 10 minutes. And we just leave it here for 30 seconds. Before that, I will try to wipe any acetone or methanol in the slide, in the well. You don't have to touch the tissue paper with the well or the slide. Just invert it a little, and acetone has a tendency to evaporate. So we leave it for 30 seconds and acetone will evaporate and all the acetone will be removed from the slide. Just wipe some from here, the edges. Okay. So now the wells are dry and acetone has evaporated. That's what we are thinking. And now we will rehydrate the cells. Since you already know, Rashmi told that the acetone and methanol, they do dehydration slide. So to rehydrate the cells, we will expose them again to 1x PBS. So 3 times wash with 1x PBS again. First wash. Touch with just the edge. And the second wash. I hope you are able to clearly see how that droplet is going away after using this vacuum suction. Remove all the residual PBS if there's near the edge of the well or near the well. Make sure you just touch the edge of the well with this suction pump pipe, because if you touch it inside the well, your cells might also get removed. So the cell density and of the cell pellet, it will be considerably low and you won't be able to image what you are deciding. So we have washed it 3 times with 1x PBS, and now we'll add 20 microliter of blocking solution to this well. Now, what is the purpose of using blocking solution?
Actually, blocking solution contains BSA, we all know, which is bovine serum albumin. It is used to prevent non-specific binding of the antibodies, which we'll be using further, and it will prevent the binding at these sites, and also it will prevent the background noise by blocking the antibody non-targeted sites.
I have added the blocking solution, and this is the bottle of BSA, bovine serum albumin, that we have used. We have used this BSA to prepare this blocking solution here. And so, right, we have added the blocking solution. It is advised to wait for 15 minutes, leave this blocking solution here, and then wash again with 1x PBS. This washing step is important to remove any residual chemical that you have exposed your cells to. So I'm removing the blocking solution first. Tips. So I'll get the tips. This is 1x PBS. First wash. Wet. Second wash. And then third wash, the final wash. I'm removing all the residual droplets from the sides and the edges of this well. Now, it's time for the actual thing. Now we add 20 microliter of primary antibody to this well. Now, what primary antibody does, we take the primary antibody specific to our protein, which we want to visualize. So, for example, if we want to see the tubulin, so we will take antitubulin antibody. Similarly, if whatever your protein is that you want to visualize, you use the antibody specific to that protein, and the secondary antibody, it is specific to the
It will bind to the primary antibody.
it will bind to the primary antibody. So it is usually anti-rat, anti-goat, such kind of antibodies.
Also, the secondary antibody will be conjugated to a fluorophore, which you will be able to see in the fluorescent microscope, and that signal signifies that there is your protein.
So we treat it with the primary antibody, leave it for 1 hour in the humidified chamber. I'll quickly show you how we prepare the humidified chamber. So just take a tip box. So I'll use this box. I'll use this box, just remove this tray. I'll fill it with the tissue papers. Fully fill it with the tissue papers. If you still feel there's any gap or any light can come from anywhere, you fill it with more tissue papers so that there can be lots of water inside this box. And your slide should not be dry at any point. So I'll add water to it.
And please remember, whenever you are adding antibodies, and this should be done in the dark.
I've just sprayed some little water to it. Just don't add a lot of or a glass full of cup full of water. I've just sprayed a little water on this tissue paper that we have kept. Now, when we treated our slide with the primary antibody, we keep it in this box. We cover it with another set of tissue papers on top. Don't let it touch to your slide. So add some water to this as well, and your humidified chamber is ready. Just close it and keep it in a dark space so that there's no light that can enter to your antibody. And you leave it in this humidified chamber at room temperature for 1 hour. Then after 1 hour of incubation, we treat these wells with PBS again 3 times. Now you know why, because to remove any residual primary antibody that is in this well, we treat this again with the 1x PBS. And so after this, we add the secondary antibody. Same again, put this in this humidified chamber and leave it for 1 hour of incubation. And so this time is standardized. This 1 hour keeping 1 hour incubation time, it has been standardized. If you leave it for too long, then there might be non-specific binding inside your cell, and you won't get the accurate or good signal. You won't be able to quantify it. So it's advised to just leave it for 1 hour or maybe maximum 1.5 hours, not more than that. And we treat it with a secondary antibody, put it for incubation for 1 hour, and then wash it again with PBS. Now, in the final step, in the last step, so you have added your secondary antibody. Now you are ready for the mounting medium.
So it is kept at 4 degrees.
It is kept in 4 degrees. This is the mounting media we use. This is the Vecta Shield media, and it already contains DAPI. So you don't have to worry about staining your nucleus with DAPI externally. This already contains DAPI. There are actually two types of Vecta Shields, with DAPI and without DAPI. You can use any one. But in our lab, we generally use the one which is stained with DAPI. So, how much to use? 20 microliter? No, please don't. This is very expensive, so just use very small amount. I will use 4 microliters of this only. Um, Generally, it is advised to use just a small drop uh, of this vector sheet. So 4 microliter is enough for your cells to completely cover with the mounting media. If you still feel there's uh, space left and your cells are not covered, just add one more drop, not more than that. So I'll immediately keep this back in 4 degrees. Now we leave it for some time for the mounting media to settle in and for your cells to be stained with DAPI. And we squeeze out. Okay, so now this is all the treatment. All the treatment steps have been done here. We have treated with primary antibody, secondary antibody. We have first sliced the cells, fix the cells first, then spheroplastid in, fix the cells first, then prepare the spheroplastic cells by lysing the cell wall of the E cells. So when you are using E cells, it is important to lyse the cell wall. When you're using other cells, this lysis part is not required. And we have treated it with primary antibody. No, first methanol and acetone to permeabilize the cell and treated with primary antibody, secondary antibody, and then put the mounting media. Now, in the final step, we will cover this slide with a cover slip because we generally visualize this in the inverted microscope. So this is a very thick slide, and to this is a very thick slide. See, so to visualize this in the microscope, you won't get a very clear picture of your cells at 100x magnification. So we use the cover slip to cover this slide. I'll get the cover slip and we cover it with the nail polish. So I'll bring both of those things. If you have any questions, please feel free to ask them now. You can write it in the chat box or unmute yourself. Feel free to ask.
Usually after adding the secondary antibody, all the steps are done in dark, but because we have to demonstrate you, that's why we are doing it in the light.
So this is the Corning cover slip. See, this is the Corning cover slip. Its thickness is 1.5 mm. And we use this cover slip to observe our cells in the Leica microscope. Here, kept it here. And I'll wipe this slide fully with the tissue paper. It is advised to use Kim wipes here so that you are sensitive with the cover slip and you don't break it or scratch it in any sense. Now we have wiped the cover slip. Now this step is important. How to seal it with the nail paint. So this is any normal general transparent nail paint. You put it here on the edges, just on the edges.
We can't use the colored nail paint.
Try to keep the nail paint as much away from your well as possible. Just the edges. I've not treated these wells with the cell, so I'll just do it quickly. Away from my well. I'll keep the cover slip. I'll get the cover slip here and I'll push it a little so that there are no bubbles left inside any well. If there will be bubbles inside your well in which you have your cells, then you'll
will
have trouble
hinder.
imaging. Cover slip, and I'll seal it again on the edges. You need to seal all the edges properly. So our slide is ready for imaging in the microscope. You can visualize this in 100x magnification with the oil immersion. And you will be able to see your E cells and the protein that you have tagged, and with whichever fluorophore you've tagged it. So you have to check it in that particular channel. You must have already learned it in the different lectures of this course only. So if we will visualize this slide in the microscope, let's show you how it will look like. And
We can keep it at -20 if we are not visualizing it.
yeah. You can keep it at -20 for some time. For some time, you can even keep it at 4 degrees. But for longer periods, you can keep it at -20, store it for some time, and you
But
can re-image it.
preferably image it next day only. Otherwise, you can keep it at the -20 also.
So if you're standardizing, if you are standardizing the protocol, then do it as per said. Stick to the protocol. Don't leave it for a very long time. Don't freeze the cells for a very long time. Just strictly stick to the protocol. If you have already standardized, and if you know at what conditions, at what temperature, and for how long your antibody takes to bind, then you're good to go. So now we'll show you how the cells look like if we treat our cells, cerebroblast cells, with antitubulin antibody, and we'll tell you which secondary antibody we have used.
So, am I audible through this? Or we can unmute through phone only? Okay. So, I will show you. Okay. This is how the immunofluorescence slide will look in the microscope. This you can see these blue dots, these are the nucleus inside the cells, and this is stained with DAPI, this nucleus, and this red is the tubulin. So we have used anti-tubulin antibody, and this using the TRITC conjugation that gives you the red color. So if you're planning to do multiple color imaging, then you can see how it looks like. Here in the green channel, you can see that the Recate protein is visualized using Elexaflor 488 conjugated secondary antibody, which will give you green color. And the primary antibody will be against the Recate, and that will have the HA tag. The secondary antibody will be against the HA tag and conjugated to Alexa 488. The blue channel will be your nucleus, and this tubulin, anti-tubulin antibody, the secondary antibody binding to primary antibody, which is anti-tubulin, and secondary antibody is conjugated with rhodamine. That gives you the or TRITC, that gives you the red color. So
these are the merged images.
So these are the merged images. You can see the metaphase cells because the tubulin is short. Anaphase cells, you can see longer tubulin. And similarly, using the tubulin imaging, you can categorize your cells into different stages of the of your mitosis or meiosis. So what else? I think we have showed you that image also, how spheroplastic cells look like. I'll show you again. Yes. So I have showed this earlier, maybe if someone has missed it. These are the non-spheroplastic yeast cells and these are the spheroplastic yeast cells. So if anyone have any question, they can ask in the chat box.
We already have these images of the immunofluorescence on our lab's website.
Yeah.
You can go to Mehta Lab IITH and visualize these cells. And if you have any questions, you can email us or ask through different contact channels, which are present on the website only.
Okay. Yeah. So yeah, I don't see. So there are no questions. I think antibody selection we have learned in the class. Sir told in the previous classes that when whatever protein you want to visualize through immunofluorescence, so primary antibody will target that protein. And there is one website called Jackson Immunoresearch. So for that website you can visit to choose your secondary antibody. It gives you variety of options. Sir has demonstrated this in the class, and it gives you a variety of options to choose your secondary antibody, and there are options also to choose for what color you would want with your secondary antibody. So I think we are done with this demonstration. Anything else left, Kirti?
No, I think we're actually done with the demonstration. If anyone has any questions, you can ask now or write it to us. We'll wait for 10 minutes before ending the demonstration. Please ask any questions, any doubts, and you can also send us an email if you have any question later. Please feel free to contact.
Okay, but there are no questions, so I'm thinking to demonstrate that Jackson website.
We'll demonstrate how you should select a secondary antibody. Usually, this part is difficult for a person who is just planning their research and they're planning the experiment. So, this part is very important.
So this is the website, Jackson Immunoresearch, for choosing your secondary antibodies. So go.
Okay, I'll summarize everything that we have done in 5 minutes quickly. Just look at the website for the selection of secondary antibody first.
So if you see here, you can choose, like suppose we want whole IgG antibodies. Why whole? Because this will give you more robust signal. So whole IgG antibody. Then target. So what target, depending on the antibody, what target do you want? Suppose your primary antibody was developed in mouse, so you choose the target accordingly. Then host organism. in which it was developed. And specificity, you can select. If you want the robust signal, you can choose H plus L. If you want any FC receptor specifics, if you're studying any FC receptor specific function, you can choose IgG, FCC. So this will vary according to your demand. So suppose I do this, like I wanted to coat, and if you want a, and this, suppose I want
Rabbit.
rabbit.
This is rabbit anti-goat antibody.
Yes. And I want this one. Select the conjugates. So these conjugates are available with the type of antibodies I want. In the green channel, if you want to visualize, it has this and Alexa 488 and FITC. Suppose I choose Alexa 488. Go to results. So see, even after selecting, giving so many specifications, there are two antibodies available. And this one tells you that it has been absorbs and it has minimum cross reactivity with this humans and human serum proteins. This is how you choose your secondary antibody using this Jackson Immuno Research Lab. If you have any questions, you can ask also, and even this will be demonstrated, or I think was demonstrated in the class. Yeah. So someone was asking us to summarize the topic, right?
Yes. Yeah. So I will quickly summarize it.
Yeah.
Meanwhile, others can ask any questions if they have.
Yes.
Okay. So I'll show you if someone has joined late. So I'll show. This was the setup. Actually, this is the protocol that we used for performing the immunofluorescence experiment. This is the vacuum pump, and we usually keep our spheroplastid cells and enzyme here. So I'll summarize you what we did and what we used. So we have grown our cells to log phase. We inoculated the primary culture to 25 or 30 ml of YPT medium, and we grew them to 1.2 OD. So these are the 1.2 OD cells, and we took 3 or 5 ml of culture. We took 5 ml. So we took 5 ml of culture in this 15 ml Falcon. And we have treated it with 37% of formaldehyde to make it the final concentration of 4%. Now, okay, so this formaldehyde fixation step is done to fix the cells, to stop the cells at whatever stage they are present. So to prevent them from dying and to move them from to any further stage, we fix them at a particular stage. And then this was incubated at room temperature.
Also, do not forget to mix it well before incubation.
Yeah, it's very important to mix it. And then after 2 hours of incubation step, after 2 hours of fixation step, we resuspend them in 1x PBS. So PBS, it is a buffer here to maintain the osmotic pressure. And then we added the spheroplastic solution. The spheroplastic solution has a sorbitol, so it maintains the osmotic pressure and prevents the cell from bursting when you add zymolase.
Okay, let's just adding one thing. Whenever you are doing the washing step, it is just done to remove the previous reagent.
Yes. Yes. So the washing step here, you will observe in the entire protocol. We in the entire experiment, we did washing step with 1x PBS only. None of the washing was done with water. And so spheroplastic solution was added. After that, we added 10% beta mercaptoethanol and we added zymolase. So beta mercaptoethanol, it created the reducing environment for the zymolase to work well. And the zymolase here, it is an enzyme to break the cell wall of the yeast, which is generally difficult to break. So for anything to enter inside the cell, it is very important for to make the cell permeable and to remove the cell wall, which we did with the Zymolase treatment.
All of these regions are in a way helping to make the cells spheroplast to break the cell wall.
Yes, and we already showed how spheroplastic cells look like. And then we use this 10 well plate. I'll show you again. This is our 10 well plate for immunofluorescence experiment. These are the 10 wells. This is coated with Teflon, and we put poly L lysine to this well. You can use different strains in these 10 wells. In whichever well you have to add your spheroplastic cells, just put poly L lysine. It is an adhering agent. So poly L lysine will prepare a surface where your cells, spheroplastic cells, can attach to. And then you wash it with PBS 3 times. You remove the poly L lysine. First of all, add it, keep it for 10 minutes or maybe 15 minutes, and then
Aspirate it off.
aspirate it off, and then wash that particular well with 1x PBS, this 1x PBS for 3 times. And then you should put that poly L lysine treated well plate in 2 bottles. First, dip it in methanol, leave it for 5 minutes, then dip it in acetone and leave it for 5
Do not
minutes.
forget that those bottles should be previously incubated at -20 and should be pre-chilled.
So you have to keep the bottles in -20 only. Keep the slide in the bottle in -20, keeping it in -20. This incubation step will be done in -20 only. And so methanol and acetone will permeabilize the cell, and these are the actually fixatives. So they will dehydrate and permeabilize the cell wall for the primary antibody to enter. And so after the acetone treatment, wipe the acetone from your well plate, not by touching the well. The well which is containing your spheroplastic cells, do not touch anything, neither the tip, neither the tip of this vacuum pump, not even your hands and the tissue paper to that well, and just aspirate everything from the edges. Now let the acetone to dry out, and wash it with 1x PBS 3 times.
Just to rehydrate the cells.
Yes, just to rehydrate the cells, because the methanol and acetone would have removed all the water that's present in the media or the surroundings. They would have removed all of it.
So you have to make sure that the wells do not dry out.
Yes. And then after that, we add the blocking solution. This contains BSA. This is the bovine serum albumin, BSA. And we wash it with a blocking solution 3 times again. And wait. We put blocking solution. We wait for 15 minutes. And then after 15 minutes, you aspirate off all the blocking solution and wash it with 1x PBS now 3 times. Now add 20 microliter of primary antibody. Leave it for 1 hour in the humidified chamber that you have prepared with the tissue paper and added water to it. And then wash it with 1x PBS, then secondary antibody, and then wash it with 1x PBS. We have used the mounting solution, the Vecta Shield mounting solution, which already contains DAPI, so you don't need to stain your nucleus externally. And covered the glass slide with the cover slip and fixed it with the
nail polish.
So that's done. This is the immunofluorescence experiment. We have demonstrated at each and every 7. I hope all of you understood it. If you have any questions, please ask. I don't think we have enough time, so you can email us and ask anything.
Yeah. Okay, so I think we are done for this live demonstration. So thank you everyone for joining.